I know that. Tracy told me. Anyway, welcome to our kickoff meeting for the 2018-19 Garden Club year. 2019 is our 50th anniversary, so this is a big year for the club. I wanted to thank our hospitality committee, which is Bev Shapiro, who arranges for this venue for us, Janice Cook, who isn't here tonight, and Jenny Johnson, who put this on this evening for us. Thank you, Jenny. Please continue to enjoy the refreshments and while we go through our meeting. A couple of announcements, but I don't want to take up too much time. First of all, our orchid level sponsor for the Garden Club is this gentleman, Mike, Renick, and Eric in the front row. Eric's. This year, to coincide with our 50th anniversary, they are upping their historic giving level of $12,000 per year, and they're upping that to $14,000 this year. To have their total giving $50,000 for our 50th year. So that's very special to us, and thank you, Mike and Eric.
Thank you.
So Mike tonight is recording this meeting, and we have a Facebook page. The Garden Club has a Facebook page. So now we'll have this meeting for anyone who couldn't attend and is interested in our speaker. They can go to the Longboat Garden Club Facebook page and watch this meeting, which is pretty cool. So thank you for that. And Joanne Schwinke maintains our Facebook page for us. I didn't want to get too bogged down with our upcoming programs, especially since I put my glasses up here. Our next event is the Welcome Back Luncheon. We will take a few minutes for Linda to talk about that, but all of our other meetings, programs, anyone who's a member, and all of you who are here who aren't currently members, I know you're future members, so you'll be getting one of these yearbooks. And on pages 4 and 5, every one of our events and our fundraisers, our programs are listed in here, with the exception of our field trips that are amazing and coordinated by Barbara Kerwin. All of those are covered in our monthly newsletter. So please, when you get a newsletter from the Longboat Garden Club, take a minute and read it, because there's always something engaging in there that would likely interest you. Just a few of the events, though, that you're certainly not going to want to miss. The holiday party, Linda Ulrich is also on that committee. The Taste of the Keys and Fashion Show, Lynn Haycock is here. If you have questions about that, you can get with her later on that. The Valentine's Day event is also Linda Ulrich and Pat Ventillo and Susan LaPrete, and I know Susan is here as well. Y'all can get with them. The Home and Garden Tour, are you here? Kathy Bishop and Marlene are here. If you have homes that you'd like to place on the home tour or ideas of homes, get with them. And so for that, now, Linda, if you want to talk just a moment about the Welcome Back Luncheon.
I have forms here for anybody who'd like to fill it out and pay us today, but we are having the Welcome Back lunch this year at the Holiday Inn Lido Beach at the Sand Dollar Dining Room, which is on the second floor with a beautiful view. And so it is $30, which is pretty similar to what it was last year. And it's on November 7th, Wednesday, November 7th. For all the golfers who couldn't make the lunches in the past, it's on Wednesday. So please see myself or Susan LaPrete and sign up. And if you don't have your money or your form today, you can send it into the mailbox. We look forward to seeing you all.
Okay, thank you, Linda. I did get a lovely video from Tracy Fanara. She was called away unexpectedly-ish to a film festival in New York City for some of her work. And she expected or hoped to be back in time, but was obviously not. But she assured me that she brought in someone even more brilliant than herself, which is a big sell. But she also, in her video that she had hoped we could show, made specific comment of the fact that she was a scholarship, a Longboat Key Garden Club scholarship recipient, and how that helped fulfill her career path and changed her life. And she said, I want all of you to know that what you're doing is important and it's very special, and that we do indeed change lives through what we do with our fundraising and giving. So she specifically wanted that shared, and so since I can't share the video, I wanted to tell you that.
Hi everyone, my name is Dr. Tracy Fanara and I am the Program Manager of Environmental Health at Mote Marine Laboratory. I am supposed to be there tonight, but I am in New York City doing a science and film festival, which is far outside my artsy comfort zone. However, you are in really good hands. The best person in probably the world to speak on red tide is Dr. Vincent Lovko, and he is the one filling in for me tonight. Thank you, Vince. I really, really appreciate it. I was a Longboat Key Garden Club scholarship recipient, and because of that scholarship, I was able to graduate on time. It really changed the path of my career, and I just wanted to let you guys know that what you're doing is amazing, and it really does change lives. And thank you. Have fun. Vince is awesome.
So, our speaker this evening is Vince Lovko, and you are the...
Sure. So I'm a scientist at Mote Marine Lab. I run the Phytoplankton ecology program. Does anyone who knows what phytoplankton means? Good. We'll talk a little bit about phytoplankton and red tides, where red tide fits into that. So I've been at Mote for about 8 years, working on red tide the whole time, of course. I have a few other small projects in my program, but for the most part, we work on red tide. A lot of that is state funded. So tonight, I don't have a formal presentation. I actually just got a text from Tracy last night letting me know that I'm the one on call for filling in for her. But you know, red tide, I've been talking about it, well, for years, but pretty consistently for months. So I think we can handle this. So I'm going to keep it a little loose and kind of just talk a little bit about red tide and put it into some context, but then quickly, open up to questions or maybe even ask you some questions and let that spur the discussion. Because I'm sure a lot of you have some thoughts on your minds about red tide and would like to hear what a scientist has to say about it. So, one of the things about science, and the reason that I got into science, is because a lot of it's problem solving. A lot of it's kind of like a puzzle. And that's what the analogies I started using recently to look at red tide. Red tide is like a big puzzle. And it's a really big puzzle with many, many pieces, and maybe it's even multi-dimensional. But and so a lot of people want to know, okay, well, what's the deal with red tide? Why does it happen? Why do we see it? Where we see it? When we see it? What can we do about it? Well, all those get answered by filling in that puzzle, filling in those pieces of the puzzle. And sometimes we'll find a piece and was like, I think this goes in this puzzle, but we don't quite know where it goes. Or maybe sometimes we find a few pieces that fit together and we're certain that's part of the red tide story, but we don't kind of know where within that puzzle it goes. Other times a little piece of the puzzle, little section starts to take form. And so we do learn things along the way, and but there's still a lot of that puzzle that's missing. So we're still working on it. We're still trying to find a lot of things out. So back to the phytoplankton thing. So this is a softball, so this will be an easy one. So phytoplankton, are they good or bad? Both. They're mostly good. They're mostly good. So phytoplankton, in the oceans, phytoplankton are the base of the food web. Things eat phytoplankton, other things eat those things, and eventually you get fish and all the other things that eat fish, and including us. Of course, we're not tied to only eating fish, but if we didn't have phytoplankton, we wouldn't have anything else in the ocean. So that phytoplankton themselves are good. Occasionally you have them do bad things, and those bad things, what we call harmful algal blooms, can take a few different forms. They can be harmful because just the sheer biomass, they get a lot of nutrients and they bloom, and they're not necessarily harmful in the sense of producing a toxin, but they bung up the ecosystem. They block out sunlight, they use up oxygen, and they cause bad effects that way. Other times they might have mechanical effects, so if they have spines and things on them, and they're microscopic, they're little tiny things, but they have spines and things on them, so when they're in high numbers, they can pass across fish gills and tear up the fish gills, and that can kill fish. And then of course there's the ones that we're familiar with here, the toxin-producing ones that can produce a variety of toxins. Well, all the different harmful algae that are toxin producers around the world produce a variety of toxins, and those can cause a lot of problems. They can kill fish, they can make humans sick, and kill a lot of other animals. And that's of course what we're dealing with here in Florida. So our recent red tide, who thinks it's the worst red tide that we've ever had? Okay, and it could be. It's really difficult to say. A lot of that depends on how you define worse. Is it the largest in spatial area that it covers, and even then, for how long, for how much time? Or is it just the overall duration, how long it lasts? We're on about 12 months with this one. In the past, we've actually had blooms that have been as long as 20 months. Fairly recently, we've had 2004, 5, and 6, we had an 18-month long bloom. More recently, we've had maybe 6 to 9 month blooms. And those blooms, though, are the tail end of those blooms. The core of it might be about 4 to 5 months, and then they trail on. A lot of times they get stuck in Sarasota Bay, just kind of hang out there for a bit. But generally speaking, we are on a long bloom. But we've had 18 and 20 month long blooms in the past. And then there's the effects. Maybe the effects are what makes it a really bad bloom. We've had, I can't even keep up with the numbers myself anymore, but a large number, we'll just leave it at that, of dead manatee, turtles, dolphins, and of course, loads and loads of fish that have been scooped off the beaches, and other, many more dead fish that have not been scooped up off the beaches because they sunk out off the coast. And of course, when they sink off the coast and all the other dead things sink off the coast, including the bloom itself, then it starts to use up oxygen, and we've had what is referred to as dead zones starting to show up. Probably temporary dead zones, I'm sure. They won't be a seasonal reoccurrence like they are on the North Gulf Coast. All right, so this bloom, currently, we have a bloom on the Panhandle. And if there's one slide I wish I could show you, is a composite that I made today. So who thinks the hurricane might have helped dissipate the bloom? Does anyone think the hurricane might have helped dissipate the bloom? Well, I made a composite of, who knows what satellite imagery is? Everybody understands satellite imagery? So satellite imagery is one of those great tools that we can use to track blooms. So basically what the satellites are doing, they're called the remote sensors. And what they're doing is looking at the radiation, and so there's all sorts of forms of radiation going from radio waves to gamma waves, and in there is our visible light, the things that allow us to see, the wavelengths that are bouncing off things that come to our eyes and allow us to see things. Well, some of those wavelengths are used to try to look for things in the ocean. Ocean color is what we call it. So sunlight and infrared radiation hits the water, it interacts with things in the water, some of it bounces back, and these remote sensors pick that up. And then you can use algorithms to try to determine what's in the water. So other people much smarter than myself have developed algorithms that allow us to see Karenia brevis, or what we think is Karenia brevis. So some of those satellite images, I was working with them today, and I was looking at October 6th, which is just before the hurricane passed through. And there was evidence of a bloom along the Panhandle. There's the large bloom we still have, even though it's been offshore a little bit, along the West Florida shelf, a little bit in the Keys, and then some even on the East Coast. Everybody realizes it's on the East Coast too now, right? And then I put together a bunch of images from the 13th, which is just this past Sunday after the hurricane came through. What do you think I saw? I saw a bloom on the Panhandle, even the Panhandle, which that surprised me even. Although, I say suspected bloom, because until we can go out and take a sample and confirm what's in it, we don't know for sure. But what appears to be a bloom on the Panhandle, or bloom on the West Florida coast, and in the Keys, and some evidence still on the East Coast. So what would you say about if somebody asked you, do you think a hurricane gets rid of red tide? And we get asked that question all the time, and it's a difficult thing to answer because how often do we have a hurricane come through while we have a red tide? In 2012, the first red tide I experienced in 2012, I got to Moat in 2010. In September, we started to see some increasing cells and you know, a little cautious, like something might be forming. And then the remnants, I can't remember the name of the hurricane, but the remnants of a hurricane came through. We thought that might stir up the water enough to dissipate the bloom and wash it away, but it didn't. It reformed after the remnants of the storm passed, and we had one of the worst, at that time, one of the worst blooms we had had in a while. Lasted into January and February. We had record manatee deaths from that one. So, but to put that into context, you know, we have had some major blooms. In the 90s, we had a bloom that made it all the way up to Jacksonville, had really bad effects, killed fish, made people sick, you know, the respiratory irritation. In 1987, we had a bloom that actually made it all the way up to the Carolinas. I can't remember if it was only South Carolina, if it actually made it up to North Carolina, but made it up to the Carolinas with enough oomph to actually cause problems and kill fish. So this is certainly not the first time a bloom has made it onto the East Coast. Does anyone know? So, all right, so here's a question for you. So the bloom on the Panhandle, the bloom we have on our coast here, and the bloom on the East Coast, do you think they're all the same bloom, or are they different blooms? It's a little bit of a trick question because we can only, you know, say what we know without a good genetic marker to be able to tell different populations. We couldn't really say for sure, but we can, based on what we've seen in the past, we can make some inferences. And so basically, the bloom that we've had on our coast, what we call the West Florida Shelf, we've experienced a lot of northern winds and through, I guess, July and August, and that was really starting to stretch that bloom up north. And so some of that probably got entrained in some currents and made its way to the Panhandle. And then and that actually happens quite a bit. So the East Coast thing is a little bit more rare. But what happens, so we've had a very intense bloom along the West Florida Shelf going into the Keys. And so there's a current called the loop current. Does anybody know what the loop current is? Of course you do, Lenny. So the loop current is a current that comes in between the Yucatan Peninsula and Cuba. It kind of curves into the Gulf of Mexico and then curves around to the east and then comes back out through the Florida Straits, which is that area of water right below the Keys, then makes its way up the east coast of Florida and then eventually merges with the Gulf Stream. So what happens is that loop current can kind of, some of the eddies and some of the currents associated with that can pull some of the bloom out into that Florida current. And that's most likely what happened. And we are seeing evidence of cell counts all along Miami and all the way up to, so far St. Lucie County, I think, is the northern extent. Hopefully it won't go any further than that. All right, let's see. So I'm gonna open it up to a question. I'm gonna see who's got the biggest. Yes.
How far north have you ever been able to track it up the East Coast of the United States?
Well, like I said, in 1987, there was a severe bloom, and it made it out, and actually, so it goes into the Florida current and then merges with the Gulf Stream, and it did that particular year and made it all the way up to the Carolinas. To my knowledge, that's the farthest north that it's been seen. I saw that one first.
Question, would you eat any fish today?
You know, I got so on Thursday I gave a talk at a realty event at an open house, and someone asked me about stone crabs. And well, you know, stone crabs are so good. Well, to a large degree, commercially available fish, shellfish, crabs, you know, crustaceans are considered safe to eat. There's a lot of regulatory things in place. Now, catching it yourself, when it comes to fish, generally speaking, what we've always gone by is if you fillet the fish, you should be fine. Well, I say should, right? I don't want to hear that. I want to hear that I'll be fine. Well, you probably will be fine. See, as scientists, we always speak in these caveats. So, generally speaking, it was always thought that the toxins are going to be in what the organism eats. And so, therefore, it's going to be in the gut. So, if you don't eat fish guts, you're going to be okay. And that those toxins get metabolized very quickly, so that the toxins aren't really available to get into the muscles and other tissues of the fish. And that is true. However, there have been some researchers somewhat recently that have found some metabolites of the toxin. So the toxin, as it starts to get broken down by the body, they have identified some in the flesh of tissues, or the tissues of fish, the flesh, the muscle, the part that we eat. But what we don't know is if those were actually toxic. And because we don't see it very often, we don't see it often enough. And then there's a lot of unknowns. I've been recently studying the effects on stone crab. There's a colleague of mine at Moat, he works on crabs and other crustaceans, and So there was anecdotal information that stone crabs, I think it was the year before last, we had a small red tide, relatively small compared to this one at least. And the stone crabbers were noticing that the crabs weren't looking right. They were lethargic, they were kind of droopy, they were discolored. And so we wondered if that had to do with the red tide or maybe just something like low oxygen. So we actually put together a study, and very simple, we put crabs with red tide, and it increased the mortality significantly. And we actually published that, and then we went on to work with larval stone crab, and some trophic transfer studies, because stone crab eat, a lot of what they eat are clams, where clams are filter feeders, and filter feeders build up the toxins by building up the cells. So if something eats a clam that has built up all these cells and toxins, then they get that blast of toxins. And one of the things we don't know about crabs is does the toxin get into their muscles? Because they have very, very different muscle structure than a fish. And that's something we don't know yet, but we're actually literally testing that well, maybe not as we speak. I think everybody's left the lab by now, but we're testing that now. Literally, we're testing that now, and hopefully we'll have some answers pretty soon. All right, so what about, yes?
So what causes it? And then when it goes away, does it really go away, or does it just move somewhere else?
All right, so that's an excellent question, a very complicated question with lots of different answer parts, so that'll launch me into a little bit of a whatever you want to call it.
Let me ask you.
A diatribe. Yes.
If you had all the resources you need, can you fix it?
That's a good question too. The short answer is probably not. Probably not. And if I could show you that image of the bloom on the Panhandle, the bloom on our coast, the bloom in the Keys, the bloom on the East Coast, you just realize the immensity of it. Now, who has heard that? Who I'm sure you have, and you probably heard me say it on the news or whatever. But who has heard that red tide is natural, naturally occurring? Who doesn't believe that? That's okay. No, don't be shy. It's all good.
A lot of people
Well,
say it's not, it's caused by what
Well,
we're doing.
so the key here is cause, and that's an important distinction. So as scientists, when we hear cause, we think the initiation, what causes a bloom to first start to happen. And what you might mean by cause is what causes us to have a big, massive, very intense and serious bloom that we're having now, and especially all the effects that it's having. Well, so first of all, let's back up a little bit. I do believe that it's natural. I think it's not only the organism itself, but I think the event of red tide is natural, or has occurred here for a very long time. I'm sure you've heard the whole Spanish explorer thing, right? Well, there's a Spanish explorer. It's a Spanish name, and I can't pronounce it very well. I do have it written on a slide, but Alves de Souza, I think, is his name. So he was a Spanish explorer back in the 1500s. And he came into the Gulf of Mexico, and he got a lot of stories from the Native Americans. They suffered great tragedies, that whole expedition. I recently bought the book that this stuff is printed in. And he got the stories from the Native Americans, and they would tell him about this red water. Sometimes the water is red, and when it's red, you can't eat the shellfish. And there's fish deaths associated with this, and I think in some cases birds as well. Can we say for sure that was red tide, the red tide that we know? No, of course not. There's no samples hanging around from back then. No one had glutaraldehyde to preserve a sample for however many hundreds of years. So we don't know, but there's a lot of evidence that suggests that it is. Now what we do have is recorded evidence from as early as 1844 of these events, like scientifically documented evidence of these red water events, as they called them at the time, associated with fish kills, associated with the irritating fumes, as they called it back then. And that persisted for a long time, for about 100 years before we finally identified the organism. It was in 1947, a very bad bloom that we had in 1947, that spurred a lot of things. And one of the things is there were no scientists here in the Tampa Bay area that were working on red tide. They brought somebody up from Miami, and they brought somebody down from Woods Hole. And they took the water, they tested it, they did a very good scientific method. Some of the methods were a little questionable back then. They would actually inject, spray some of the water into people's noses to see if that caused the irritation. This isn't a report. This isn't an actual report from the federal report. And this report also said about how people, citizens, would take buckets or pots of the red water and put it on their stove and inhale over it and get the irritating. I call that an early form of citizen science, but nothing I would recommend doing now. But anyways, they went through the scientific method. They tested it on fish. They tested it on people in ways that we certainly couldn't do now. And they confirmed that this red water was causing those issues, both the dead fish and the irritation and so forth. And then they also identified the organism. They were able to use microscopes, and they identified this unknown organism. They called it Gymnodinium breve at the time. And it's gone through a few name changes, and now we call it Karenia brevis, same organism. And so they were able to do that. They were able to find out what caused it. And then something else that also spurred a few years later in the 50s is an attempt to get rid of it. Anybody know about that? They used something called copper sulfate, a substance that's still used in some freshwater ponds to get rid of algae. And they put copper sulfate on a patch of bloom right out on the coast, and it got rid of it. It also caused it exacerbated the fish kills, because when you lyse all those cells, you release all that toxin and you kill a whole bunch more fish. And a few months later, the red tide was back, because you wouldn't be able to cover the whole entire area. So no, I don't think we could get rid of it. I think the best we can do, and now there is a possibility that we can, to some degree, minimize it. Everyone, I'll address the elephant in the room. Nutrients, everyone thinks nutrients at least makes it worse, right? That's possible. And the reason that us scientists, we don't just come out and say, yes, it does, is because that's not how science works. We don't make an assumption about what probably happens and then say that's what happens. We have to follow the data. And currently we just don't. That doesn't mean we're saying it doesn't. It means we don't have the data to say it does. But what we do know is that these things have occurred for a very long time. We've had bad blooms for a long time. And I think it was 1878 started a period of 10 years, a decade worth of blooms. Not one continuous bloom for 10 years, but 10 years of very serious blooms. So it's not anything new. And there's not necessarily one thing we can tie it to. A lot of people, of course, when the releases were occurring from Okeechobee through the Caloosahatchee back in June, they wanted to tie it to that. Well, again, those have stopped, and we had the bloom, the current bloom we have. Does anyone know when that started?
A year ago.
About a year ago, yeah. Late October, we started seeing elevated cell concentrations, October of 17. And then by November and December, it was pretty serious, but mainly to the south, which, makes sense but there were no releases from Okeechobee at that time. And then it started to dissipate in January, February. We actually thought it was going away. I thought, okay, well, this one's done for the year, that's good. And then it resurged in probably starting in March, and then through April and May, and then became really bad in June, July, August, September, October. We'll see. We have had some relief. So all this relief we had, who thinks that it kind of has gone away? Well, I guess you know that it didn't, because I told you about my satellite work today. Yes?
What clues do we have, and I mean, there may be 100,000 of these, but we don't need to hear all 100,000. What clues do we have of what may be the source, the true source of red tide? We know the nutrients, in my opinion, certainly don't help the situation. They probably do exacerbate it, but what are some of the other, what are some clues?
So our current understanding, the best that we can put together from what we can see of that puzzle, is that the blooms initiate offshore near the bottom, triggered by some event which is probably nutrient related. Now what that event is, people have hypothesized, but we don't know what that event is.
And do we know where on the ocean floor? I don't know
Well,
within a 2-mile
to give you an idea of what we don't know, all the scientists working on this, they'll tell you between 10 and 40 miles offshore.
of the U.S.
Well, of the Gulf Coast of Florida.
Gulf.
Generally thought to be, more or less parallel to the Tampa area. And so how have we found that out? What do we know about that? Well, there's been a few blooms where the collective we, I wasn't around back then, we're not studying red tide anyways, but there's been a few blooms where we've been able to go out and sample offshore and actually see higher concentrations of Karenia near the bottom offshore before we see it near shore or at the surface. So those couple of events have put that together. Also, some people have done particle modeling. So these cells are basically particles. So it's important to think of these not as like a chemical or a pollutant, that disperses evenly throughout the water. These are little particles. They're cells that can move. They can't really move outside of a current, but they can move, and they are little particles, and they get pushed around by the currents. And sometimes, so everybody's seen like the streaks of red tide in the water, right? And that usually happens because of currents, little micro currents that happen in addition to the big currents. So people have used particle modeling to figure out, okay, well, we have a bunch of particles here. Where did they likely come from? And that also points to an offshore near bottom initiation. With the overall idea that they initiate offshore, something wakes them up, causes them to start dividing, bottom currents move them towards shore, those currents encounter land, gets pushed up to the surface, those cells come to the surface where they get plenty of sunlight, they're near the coast, so they get plenty of nutrients, and then they can proliferate. So you increase those nutrients, it can make sense that you might increase the red tide, but again, we don't know how much, we don't know what role that plays. So a very important thing to remember when it comes to nutrients is, so our coastal areas in most of the world are what we call productive. Some are much more productive than others. The west coast of the US, for example, is very productive. What we mean by that is we get a lot of fish. And the reason we get a lot of fish is because we have a lot of phytoplankton. So on the west coast of Florida, it's very deep, very short shelf, and we have what's called upwelling that brings very deep Pacific Ocean water up to the surface, and that's got a lot of nutrients in it. Because everything that dies and falls down into the ocean eventually gets decomposed, and the nutrients get what we call remineralized. In other words, they get kind of liberated from their organic host, and they're easy to uptake by phytoplankton. On the west coast of Florida, we don't quite have that, but we have something similar that happens. We have a very deep gulf, same kind of things happens. Some water can get pushed up onto the shelf that can bring those nutrients. Plus, we have land. And all around the world, our coastal areas are productive often because nutrient runoff from land. And a lot of this is natural. There's a lot of things on land that decompose and release nutrients, and those nutrients go into the water. Now, anytime you increase the nutrients in any kind of aquatic system, you run the risk of kind of messing with the ecology and messing things up. So one of the reasons I'm hesitant to encourage people to think that it's nutrients making red tide worse is because what if it's not? What if you run with that banner and you get people to change things and then we find out that it has nothing to do with red tide? Then people think, well, I don't need to do this anymore. So there's plenty of reasons to reduce nutrient input into coastal systems. The blue-green algal blooms, everybody's familiar with that now, right? A few years ago, did you know about blue-green algae blooms? So the nutrient input into Lake Okeechobee is not a good thing. And that bloom is simple. In many freshwater bodies all around the country, we get those kind of blooms, those cyanobacterial blooms. And there are a lot of different species. Microcystis is one of the main ones, but there's many different species, produce many different toxins that have a lot of negative effects. They could be hepatotoxins and neurotoxins and all kinds of toxins that can cause a lot of problems. So that alone is a good reason to fight for reducing nutrient input into Lake Okeechobee. So conceptually, that solution is easy, putting nutrients in Lake Okeechobee. But then you have the political and economic battle to wage, and I'm a scientist, that's not what I do. But I would be careful about making that association with red tide. So there has been measurements of nutrients. Somebody from FGCU actually showed some data fairly recently at a panel discussion that showed fairly high amounts of nitrate, one of the very simple forms of nitrogen that plants and phytoplankton like to use, in the Caloosahatchee, and I can't remember the numbers, but they were high. And by the time they got even a mile outside of San Carlos Bay, outside of the mouth of the Caloosahatchee, they were down to pretty close to background levels. So are we putting extra nutrients into the coastal system? Yes, absolutely. What is the influence on red tide? We don't know. I think I answered the question. The source, yeah. And so, and to also address the general notion of is it natural? So one of the things that we know too, and I know this because I see it all the time, is even when we don't have red tide bloom, you take a water sample, somewhere where it's saline enough, and you're going to find Corynea brevis. You look at enough water, and I'm not talking about looking at like hundreds of gallons. You look at 1 liter, and you're going to find Corynea brevis. I pretty much guarantee it. So it's here all the time. And how that plays into our ideas of initiation, I don't know, and that's something actually we're going to try to put a project together to find out. Yes.
I was just really curious if the water temperature has anything with the increase in.
I should let Lenny get up here and talk about that.
And when the
there's been a few different ideas of water temperature. So in the literature, most of this is based in the lab. So people taking Corynea cultures, putting them under different conditions, and seeing what happens, how their growth rate is affected. And generally speaking, it's thought that in the lab, that below 16 degrees Celsius, they don't really like it very much. And above about, I think it's 30 or 31, they don't really like it. In between that, they do great. So there's this kind of zone of tolerance that a lot of organisms have to any parameter, temperature, salinity, almost anything at all. And usually there's an ideal where it's the best it can be for them and they grow really well. Then you get into this area of tolerance, and then you get to this area where they can't survive. So it's thought that outside of those two ranges, Karenia couldn't survive. Again, based on laboratory studies. Well, this summer, and so we thought that part of it is why we don't see Karenia in the summertime typically, because we usually don't, because the water's too warm. So we thought, well, the waters this summer have been quite warm, breaking some records if I'm not mistaken, or at least up there. And yet we have a very serious bloom, and we're not, so one of the things I like to caution people too is like, okay, well, surface water temperature isn't necessarily the same as the bottom water temperature. So they, that could, either that water upwelling or them going down to the bottom at night could help them survive. Well, our entire water column has been 31 for probably a few months. I'm sure it varies. So we're only, that's another thing that I think is a bit of a misconception, how much data that we actually collect. So we have a couple of main surveys, as we call them, that we do, boat-based surveys, and each of them we do every other month. So, every month we're doing one survey. We're up for 1 day on one of them, 2 days on another. And so, like our coastal survey, where we go along very near the coast, we're only about 3 miles offshore. We do that one every 8 weeks. So we get a nice little window in 1 month, and then 2 months later we get another window. What's happening in between, we have no idea. So to try to look at those trends and see what's actually happening on a regular basis, you either need more frequent sampling or you need some autonomous samplers out there, some sort of instruments that can just continually collect the data. There are some, especially for temperature. Some of the buoys have them, but they're talking one spot. So it's something I like to try to give an appreciation for, is how difficult science is in the terms of trying to collect the information that we need to answer these questions. Okay, who else has got one for me? Yes.
Are there other parts of the world affected by red tide, and if so, what are they doing to address it?
That's an excellent question. So if we think of red tide as synonymous with harmful algal blooms, yes, they occur worldwide. Different species typically. The northwest coast of the U.S. has a totally different type of organism called a diatom, Pseudonychia, that causes a lot of problems. And it really likes the fact that the ocean waters are warming up a bit. In the northeast, we have one called Alexandrium. It's a similar species or similar group to Karenia brevis. It's a dinoflagellate. In Chesapeake Bay, we have a couple of different dinoflagellates that produce toxins, and around the world we have numerous different species. Now, all of these are, just like different species of, say, a bug, a pest that you have in your house or your garden, they all react differently. They all have their own ecology, their own biology, and they don't necessarily behave or respond to the same things. So generally speaking, at least with the Alexandrium, what they try to do is get better at predicting. So Alexandrium up in the northeast, it forms cysts, and these cysts form cyst beds. And so they can go out and they can take cores and they can see where the cyst beds are for a particular season and make a prediction on how bad a bloom is going to be for that season and where it's going to be. And they've actually geared it towards now allowing the shellfish fishermen to harvest early if they think there's going to be a bad bloom. So that gets to this idea of mitigation. So these words get kind of twisted a little bit, but generally when we're responding to a hazard, we have prevention, we have control, and we have mitigation, right? So prevention, that's pretty obvious. You stop it from happening. You find out what causes it, you stop that cause, and then you don't have that hazard occur. That's very difficult to do. The other one is control. So you're doing something about the hazard. You're shortening the duration, the intensity, the effects, and that's where it kind of blurs into mitigation, but you're not necessarily getting rid of it or stopping it from happening. And then mitigation, generally you're not really doing anything about the hazard itself, but you're trying to protect people and resources from the effects of that hazard. And that's where we focus a lot of our efforts, because there's more you can do there. You can educate people. You can find out what the toxicity is in shellfish and keep people from eating toxic shellfish. You go back a few decades and people, not from Karenia brevis, but in the US, people died from eating shellfish that were contaminated with algal toxins. To our knowledge, no one has died of Karenia brevis, brevetoxin. But people have died. Now in the US, that essentially doesn't happen anymore. But another form of mitigation is prediction and forecasting, understanding when and where a bloom is going to occur. And that's something that we just haven't made a lot of progress on with Karenia brevis. Now there's a group of us at Moat, Fish and Wildlife and USF that are putting a team together to put in a proposal to address exactly that, initiation as well as termination, so what makes a bloom go away. If we understand termination, then we can predict when a bloom's going to end. So, I mean, this is a really bad bloom we're in, right? But wouldn't you like to know that was going to happen? Wouldn't that have been helpful? And wouldn't you also like to know when it's going to go away? I would. I could get caught up on everything I'm behind on. But so those two things are very important components, that initiation and termination, having a handle on them, on any hazard, anything. You want to know what causes it to start, and you want to know what makes it end. And if we know what makes it end, then maybe we can tweak that a little bit and cause it to end earlier. There's no guarantees of that, of course, but we can't really do much without knowing those two pieces. All right, what else we got? Yes. Sure.
Okay, you explained that through the satellite photography, you could see where they locate, like 10 miles in or 40 miles in, right? Okay, so if the cause of it was from what we were doing inland, so why it has this gap between the coastal area and the focus of the organism?
Well, that gets a little complicated too. So first of all, I don't necessarily think that what we are doing as far as putting nutrients into the water is necessarily the main thing making it as bad as it is. We'll see what the data says as we collect that data. But also you have to remember that the nutrients go into the water and they spread around, they dissipate. And they get used up by other things as well. So when we're talking about satellite data, what you have to remember is the satellites can only see the surface of the water, and pretty much the very surface, maybe down to a half meter, maybe a meter if the water is very clear, but if there's a lot of red tide, obviously the water is not clear, but it can only see about a half meter into the water. So what's happening underneath the water we don't know. So when we see a nice satellite image and you see all that orange and red that indicates a bloom right along the coast, we don't know what's happening in that open water, offshore of the bloom. We don't know what's happening. Now, if we could see down to the bottom, that'd be great, because then we'd be able to. There's the initiation. There it's happening, and it's moving along just like we thought it did. But unfortunately, we can't do that.
Vince?
Yes.
You almost got there, but somebody asked where it is, and you talked about it being here and there. But the Karenia brevis is only in the Gulf of Mexico.
Ah, yes, that's a great point. And I meant to address that when you were asking about the red tides around the world.
So we can compare apples to apples.
Right, our Karenia brevis is in the Gulf of Mexico. Only so far. Now, I did mention how it's gone up the East Coast a few times over the years. We have not seen any blooms that we think have initiated on the East Coast, either in Florida or elsewhere. And in Florida, we might have two populations because Texas gets blooms as well. In fact, maybe it was a month ago. Time blurs these days, but just around the time when we saw that we were having it on the Panhandle, it also popped up in Texas. That's probably a separate event. They get blooms as well that tend to occur the same normal time that we get our blooms here on the West Florida shelf. And that normal is usually we first see the expression of them at the surface in late summer into early fall. And obviously we're there again now, and we still have the bloom that started last year. That brings up a good point that I don't know if anyone's thinking that right now, but okay, is this bloom continuing, or do we have a new bloom starting? And to be honest, there is a researcher, I won't say where because I don't want to throw anyone under the bus, but there is a researcher that has claimed that he believes that it's a new bloom, kind of replacing the current bloom. But unless there's some genetic marker you can see, you can't really say that for sure. You can model it and say, well, the conditions are right to have had a bloom this year, but obviously the conditions are right because we have a bloom. All right, what else we got? This is fun.
So, I was really surprised when you said some of the major data you get every 2 months for something that has impacted our tourist industry and
Now,
islands.
on the other hand, we do cell counts, and we get a lot of cell count data. We do counts at Moat daily. We get counts from the Sarasota County weekly. We have a project that gets counts in Sarasota Bay monthly, and then in addition to our routine counts. And this is just Moat. And then Fish and Wildlife has a network of other institutions and partners that also get cell count data. But the cell count data basically tells us where it is and how high the concentrations are. That's very helpful because one of the resources, and towards the end of this I was going to mention a lot of the resources that are available, but NOAA, the National Oceanic and Atmospheric Administration, puts out a HAB forecast. They do it for the country, and of course in Florida they do it for our Florida red tide, Carenia brevis. And that forecast for respiratory irritation is based mainly on cell counts. So the cell counts are a very important part of the data for that day-to-day kind of information as to what the bloom is doing and what the hazards are to people. But that long-term information that we need to be able to answer some of these ecological questions that can confirm the initiation and why a bloom occurs at the time of year that it does and all those, that's what we need that long-term information for. And to be honest, it's because of funding. The funding goes up and down. Excellent graph that our president, Michael Crosby, president of MOPE that is, Michael Crosby likes to show when he's doing talks and outreach, is a graph that shows cell counts and then it shows funding. And you can see years where we have a really bad bloom, just after that funding goes up. You know, we're talking about state funding. I'm not throwing anyone under the bus, but state funding goes up and then we don't have a bloom for a while, it goes down, then we have a bloom. So what's often happening is it's after the bloom. You know, it's already initiated, we've had a bad bloom, it's going away, and then we see funding increase. And we're seeing that now again. And you know, that's not the way to do it, you know, because then you've spun a lot of things down when your funding has gone down, and then you have to spin them all back up again. Your equipment's old, you have to replace or repair equipment, you have to hire new people, you have to, you know, come up with, you know, update your methodologies, things change. And that's very difficult with you know, steady funding. Not to make this a funding talk, but steady funding is critical for any science. You know, you got to keep that research going so that you can collect that data that you need.
Is there any National Science Foundation funding at all?
Yeah, there's a lot of national federal agencies that fund, and actually recently the National Oceanic and Atmospheric Administration, which is a big supporter of harmful algal work. They started a harmful algal program in the late 90s. And another graph that's good fun to show is a graph of cell counts from the 1940s up to now. And what you see is it looks like we have more blooms now. But if you superimpose a graph of funding on top of that, you see that or actually it's not even a graph of funding, it's a graph of the number of samples taken. And you see that more recently, there's a huge spike, and more recently, I mean in the past couple of decades, probably since the late 90s, there's a huge increase in the number of samples that we take. And this is specific to Florida, to our red tide work. And so, you know, that graph that makes you think, see, there's a lot more blooms more recently, and there's hardly any since the 50s. Well, that's because there was hardly any sampling since the 50s, so we didn't see the bloom. We didn't know there was a bloom. And a lot of that's because of funding. So in the late 90s, NOAA started what they call EcoHab, the Ecology and Oceanography of Harmful Algal Blooms, and that brought a huge chunk of funding to harmful algal research around the country, and especially to Florida. And their funding has gone up and down over the years, but they just recently put out a call. They got money again, thank goodness, and they put out a call, and that's a lot of us are chomping at the bit to put together some good projects so we can answer some of these questions that everybody has. But it would have been nice to be able to start working on those questions, you know, 2 years ago or 4 years ago, or you know, maybe just after the 2012 bloom when we had a period of no blooms. What's different between that period of no blooms and what we've had over the past several years? Yes.
Well, talking about the funding, I'm sure you're aware that the Garden Club gives grants, and every year Mo puts in for a grant, but I don't think Mo rarely puts in for a grant for red tide. It's for
I'll talk to Tracy about that.
And maybe you could talk to whoever does the grant and ask specifically that the money be used, because they fill out a form to tell us what they're using it for. And though it may not be a great deal of money, we do it every year. And maybe it would be nice, since it affects our island, and we are the Longboat Key Garden Club, if they would put in a grant for red tide research.
I'll keep that in mind. And that reminds me too, I want to talk a bit about mitigation, so let's make sure we get there eventually. Yes?
Yeah. Just two questions. One
Okay.
is, like, how many different organizations are working on red tide in Florida, and how are we collaborating?
There's a great deal of collaboration. So pretty much everybody has worked on red tide at one point or another. Actively right now, pretty much all of them. The major ones are, of course, Moat, Florida Fish and Wildlife, USF, University of Miami, Horn Point on the other coast does it, FGCU does some red tide work, and we all collaborate. So us and Fish and Wildlife collaborate very, very tightly, and USF is often in that loop. As far as the other institutions, that usually happens through a project. Like, for example, this NOAA call that's out now. Several of us are going to get together because they like to have multi-institutional projects, because then you're taking expertise, because no one place can have expertise in everything. That's just ridiculous to think that you can. So you find the best that you can, and it's not just limited to Florida. I mean, pretty much every major institute that works on harmful algae around the country has worked on red tide. A big one is Woods Hole. Don Anderson, one of the kind of godfathers of harmful algal research. He's worked on red tide. In fact, we're partnering with him now to do some clay studies, mitigation strategy. Has anybody heard about the clay thing yet?
Yes.
What do you think of the clay thing?
No.
Yeah, the clay thing, any mitigation, anytime your control, anytime your control/mitigation study involves putting something in the environment to try to get rid of something else, that should make us all hesitant. But we also have to remember that while on the one hand we don't want the solution to be worse than the problem, we also have to recognize the benefit of reducing the problem. And we do it a lot. We put pesticides on our crops. It's all about judicious use of them, and doing the appropriate research, which of course we're going to do heavily to make sure that whatever you're using, you can never necessarily dot all your I's and cross all your T's, but to make sure whatever you're using, as much as you can, is demonstrated to be safe. And so that leads me to mitigation. Thank you. So you've probably heard, but the state has loosed up some money to do some mitigation and control strategies. And some of that is actually going to be used for the clay study. That's actually going to Woods Hole, and they're leading the clay study. But some of it's coming to Moat, and we're going to research some other compounds. And one of the criteria that we have is whatever we research is going to be in the laboratory first of all, and has to already have some EPA approval. There has to be some science behind it that shows that it's safe and not toxic to the environment. Of course, not toxic to the environment, what does that mean? Because first of all, we need it to kill Renick brevis, so it can't be completely benign. And the question is what else does it do? And so like me as a phytoplankton ecologist, one of the things I'm interested in is how does it affect the rest of the phytoplankton community? Again, phytoplankton are important. We don't want to wipe out the phytoplankton. That would be a bad thing. We regret that. So we don't want to do that. So one of the things I'll be looking at is what effect, can we find some minimum dose, some minimum concentration of whatever compound we're targeting and affect Renick brevis, but have minimal effect on the other phytoplankton. That would be ideal. All right, I know we got more, right? We're not done yet.
I heard you report that the bloom was 20 trillion gallons of water, something on the
I think it was, I don't remember. I think it was, at that point, I think it was 1.5 trillion, I want to say. Or maybe it was 15 trillion.
A lot of water.
But what it was based on was the measuring the length of the bloom at that time and the average width of the bloom, and considering that most of the cells, certainly not all of them, are in the upper meter of water. And yeah, so the idea of treating, of taking some sort of chemical, I mean, you're talking about, even let's go with 1 trillion just to do it on the low side. Even if you were to treat that water at a 1% concentration, you're still talking about a billion gallons of, 10 billion gallons of whatever it is you're going to put on this bloom. Logistically it becomes impossible. It becomes unfeasible. But the question is, could we do something where we have like an isolated bloom and we can treat that bloom? Could we maybe catch a bloom early? I actually don't think so, because what we currently understand, it's almost like a conveyor belt. That what we see of the surface expression of the bloom is being fed by cells from the bottom. So you could kill those surface cells. That doesn't mean the bloom goes away. It might just get replaced by other cells. Now, I will say that the clay strategy has been used in other countries. Some of the Asian countries, I think Korea uses a lot in their aquaculture facilities. It has been used, and it's generally thought to be, Chinese use it. It's generally thought to be benign, but we're going to make sure. We're going to determine whether we think it's benign or not. Basically, so the clay, it's very, very fine particles, and it gets mixed in a slurry and literally sprayed over the area of the bloom. And everyone's familiar with the ozone study that Mo did, right? Has everybody heard about the ozone study? We did a couple months back. Rich Pierce was leading that one. And they did it in a canal. So they found a dead-end canal, cordoned off just a small chunk of it, and use an ozonator. So it's this thing that was above ground. It pulls water out of the canal, runs it through the ozonator. It changes the oxidation reduction potential of the water, which basically kills the cells. And there's a holding tank to let the water come back to normal, and then it puts it back in the canal. Quite a process, and certainly you can imagine something like that would never work on trying to clear a large area. It was a conceptual thing, can this work? And so the clay, we're going to do something similar. We're going to find a dead-end canal and try it. That has red tide in it. If there's no red tide, it's kind of makes the whole thing pointless, and see what the effects are. So we'll curtain it off and test it. With the potential of, there is the potential, and part of this too is like there's a big push for this. There's a big push, partly by the public to do something about the red tide. They want us to do something about it. And the way I see it as a researcher, as a scientist, well, it's my obligation to find out what can be done. Beyond that, we will come up with the data. We'll do the studies, we'll show the data, and then it's up to other people to push for the regulatory components that might make it feasible or push against it.
Well, how are they doing in China? Are they doing good with it?
Yeah. Well, there's some anecdotal reports. There was one paper I read. I think this one was in Korea in an aquaculture facility. So this is still on the coast, but where they do aquaculture, they have aquaculture pens. And they were having I don't remember which species it was. One of them was one called Coclodinium. I don't remember if that was the one that came after the one that they were trying to get rid of. But they had a bad problem with the bloom. It was toxic. It was killing their fish. So they did the clay mitigation. It worked. They got rid of that species. But then the following year, another species that was not susceptible to the clay took its place. So, yeah. So again, you got to be careful. You got to make sure that the solution is not worse than the problem. But that's one anecdotal, or actually it's not anecdotal. It's in a paper. That's one example. But China does use it regularly.
I had heard that a person in Sarasota died from eating oysters.
Yeah, that I don't know. I couldn't speak to that. I don't know what the results of that were. I heard the same thing, but I don't know what the results were of any toxicity tests. What's that? What was that?
You don't eat oysters that don't in months that don't have ours. If you come from where I'm from, you don't eat oysters.
I lived on Chesapeake Bay.
You don't eat oysters in months
that
that don't have ours in them.
Yeah, I think a big reason for that is because the summer months, of course, summer is our red tide, but normally our red tide's in the winter here. But the water's warmer, so you can have more bacterial loads in the oysters. Although I also had an oysterman tell me that he thinks the May ones are the best. That was in the Chesapeake Bay, though. Anyways,
Well,
I'm not going to stop eating
This
oysters.
is more New Orleans.
I got you. All right.
Can you
Okay.
talk about the long-term effects on humans being exposed to this?
I can try. So the human health component is not my forte, but so as far as long-term effects, I can give you the easy answer. We don't know. There have been some studies that have tried to look at the long-term effects of brevetoxin, but to my knowledge, they're inconclusive. Brevetoxin's also been looked at as a potential benefit as well. I think primarily for neurodegenerative diseases such as Alzheimer's. Some clinical, well, it wasn't even clinical trials, but some evidence suggests that it could be beneficial that way. What's really interesting, especially considering what we have going on here in Florida, is some of the cyanotoxins, so the toxins produced by the blue-green algae that occurs in our freshwater bodies. Some of those toxins might actually, long-term exposure, and there's papers out on this, so I'm not just talking off the fly here, but there's papers out about this, might actually contribute to neurodegenerative diseases. And then we have red tide. Some of the components of red tide toxin might contribute to alleviating that. So in that way, you can look at, we live in the perfect area. No, I'm just kidding. That was a joke. Okay, what else we got? So one thing I did want to talk about were resources. So there's a lot of resources, and I don't just mean the ones you normally hear. There's of course the Fish and Wildlife website for and the Moat website for just general information about red tide and all that kind of stuff. There's the Fish Kill Hotline, and a lot of this you can find online. Fish Kill Hotline that's run by FWC, and they love to hear reports of dead fish. They like to get that data. That's very important information from them. So just telling somebody else about it or calling us or emailing us doesn't really help it. The best thing you could do is get on the hotline. And then at Moat, there's the beach conditions reporting system. Everybody knows that one, right? Of course. Who knows about our app? Do you know about our app? What's it called? So we have an app. So Tracy and I put this together. It's called CSIC. It's CSIC. Stands for Citizen Science Information Collaboration. And it's a great little app, and we'd really like more people to use it. And what you can do, Tracy has this idea of expanding it into all kinds of environmental sort of reporting capabilities. My main interest is the red tide layer. So on the red tide layer, you can report if you see discolored water, dead fish, and if you're experiencing respiratory irritation. That's extremely helpful to us. And it's also helpful to you because you can there's a map that you can look at, and you can toggle between respiratory irritation, dead fish, and discolored water, so you can see what's being reported where. So that helps you, so you know where it is, but it also helps me. So, you know, we talk about sensors and having more sensors out there. Well, humans can be sensors as well. So what I pictured when we developed this app is that there'd be, you know, tens of thousands of people all along the Florida coast, all reporting every day on their app, and I'd see a stream of green and red dots that lets me know where the bloom is and isn't. Well, now it's more like, you know, a handful, a little sprinkling of dots. So I'd love to see more people using that. So if you could try it out yourself and spread the word, it's available both on iTunes or whatever the Apple thing is. Obviously, I'm not an Apple guy, and also on Google Play. No ads, it's free. CSIC, C-S-I-C. So just think CSIC, but then figure out the letters that would make that happen. And so there's a lot of other more technical resources if you want to delve into that, and they're really cool, and they're available to the public. One of them is I think the NOAA HAB forecast you actually have to sign up for. But there's USF actually has a 3.5-day forecast model. So basically this is based again on particle movement. But if we have a bloom, they basically will tell you what direction it's going to go. It's a little difficult to decipher the map, but once you get used to it, it's not that hard. And that's on the USF Optical Oceanography Laboratory website. But if you look up USF HAB forecast, you'll find it. And then there's USF also on their optical oceanography site, they have the satellite maps are available. And I can get all this information to you, perhaps through Tracy, or if you email me, I'll make sure to send a whole list of these resources. But there's some technical resources that are actually really, they're kind of fun, but they're also pretty helpful to kind of get you to not only know more about what's happening with red tide, but also kind of wrap your head around it a little bit, because it could be a little daunting. And you know, what I said, the talk that I gave on last Thursday at the Realty event, you know, one of the things that I think we might need to understand is that red tide is a hazard that we have here. You know, a lot of places that you live, especially beautiful places, often have hazards. You know, it might be earthquakes, might be storms, might be fires, might be flood. And one of ours, unfortunately we have a few of those too, but one of ours is red tide. You know, if there's something we can do to minimize it, then we should certainly pursue that, but we also have to understand that it's part of our environment. All right, that seems like a good place to end, but if you have any more questions, I'll be happy to answer them. And I'll leave it to you to decide if I'm better than Tracy or not, but I don't want to hear it.
Well, thank you very much for speaking to us, and he didn't sound unprepared, right?
Well, I've been talking about this, let's see, what month is this? Yeah, I've been talking about this for a while.
And,
it seems like nonstop.
So I want to thank Kathy Garcia, our program chair, for lining up this program for tonight. And our next program is
November 20th?
Who's our speaker? Oh, that's Dr. Sally Selby Gardens. She's a senior botanist at Selby Gardens, and so she'll be speaking at our November meeting. So, and that's also in your yearbook. And if you're not a member, join, and then you'll get a yearbook, and then you'll be able to come to all of our fascinating programs.
Otherwise, they're on the website.
Yes, and they're on the website that Tori manages and Lynn's newsletter. So, welcome to the future members. I see a lot of new faces in the crowd, so thank you for coming, and thank you for our current members who brought their guests. And I hope you were engaged with our program tonight. Thank you very much.